Alright, so we're now going to be doing our blood smear task. So I have my purple top tube sample. I have microhematocrit tubes. I have some Kim wipes. I have my slides. And I also have a pipette. The way some people draw up their blood to put it on their slide is they use the microhematocrit tube to draw it out of the purple top. I find that I don't get enough sample on my slide to make an adequate slide. It ends up being short. Slides are supposed to be about 1 inch. Mine with the microhematocrit tube tend to be like ½ inch. They don't go far enough. So I like to use just a little pipette. So I'll show you how I do that too. So what I'm going to do, there are 2 ways to do a slide. One is either the handheld method where you have your thumb and your finger in the slide between and you push it away from you with your pusher slide, or the tabletop method, which is my preferred, but I will do both. So what I'm going to do is get my slide. I like to use a Kim wipe to kind of clean it off so that there's no debris on it. And I'll pull you closer so you can see a little bit better what I'm doing. So get my sample. Actually, I'm going to give this a quick couple inversions to mix it up. Okay. This sample is from a dog named Max that we saw earlier today. And I just take a very, very small amount in my pipette. And I put a small, tiny, tiny little dot. So I just squeeze it right until it barely kind of comes out of the pipette right there. And I just tap that little bit right onto my slide just to get a very small amount. So if I was doing the handheld method, I would hold, I'll put this up so you can see my face, hold it close to my body, take my other slide, this I'll wipe on my Kim wipe, then pull it back towards me right until it touches my sample, and then give it a good little push. This actually isn't a bad one, so it's about 1 inch long, which is what I like. It's got a pretty good feathered edge. It doesn't have any streaking, and it's got a nice gradient from the inside out. So the inside is where the thickest portion of the cells are concentrated. A lot of the red blood cells will be built up on top of each other. A lot of times you will read, or usually you'll mostly read from the feathered edge where the red blood cells are in a single layer. You'll get a better white blood cell count. Your platelet counts will be better seen there. Sometimes if it's thick or clumpy, a lot of times the white blood cells will get pushed towards the feathered edge. So you might get a disproportionate count there, beyond the feathered edge anyway, where it kind of trims out. So this is a pretty good slide. And then we'll do our tabletop method. I'll set this so that it's the feathered edge down, the butt end down to dry. I'll pull you closer so you can see tabletop. So I'm just going to do the same thing. Take a slide, get my Kim wipe, clean it off. Take another slide now, clean off the end. I'm just going to rest it. And I'll get my sample and just a small little dot right there. And then I'm just going to draw this back, keeping it at about a 30 to 45 degree angle until it touches. Let the blood smear across my slide. And this is kind of what you don't want. So I kind of went off track. My slide came up a little bit here. I've got some streaking there. So this would not be a good specimen slide to stain. I'm going to try for one more. And then we'll go ahead and do our dye. So I'll clean it off with my Kim wipe on my desk counter. Second one, get a little clean. Just sit there. I'm just gonna just get a little bit of our sample. draw this back. Alright, so this is a pretty good sample as well. There's a little bit of streaking over here on this edge. You can see there's kind of a break in the feathered edge there, but that's not so bad. You got a good gradient here, a good feathered edge there. It's nice rounded at the tip. It's about 1 inch long. I can estimate because the knuckle to the end of your thumb is typically approximately 1 inch. So it's about half the length of the slide, about 1 inch long, which is good. So I'm gonna sit these here, again, butt side down, over against the wall with our other samples right there. And I'm gonna let them dry for a couple minutes, and then I'll go ahead and stain. Okay, so I've let my slides dry for a few minutes so that I can go ahead and do the staining. I'm going to label them with the patient's name. It's a dog named Max, so that I can identify them. Alright, and I'm going to do this at our wet sink, which also doubles as our dental sink, so it's a little bit bigger. I have my dyes here. This light blue one is our fixative, then I have the red stain and the blue stain. This is just the dip quick step. So I am going to grab a little paper towel so I don't make too much of a mess. Uncap all of these. Get my slides out of the way. And I'm going to dip each of these into each different container 10 times for 1 second each, and I'll move from the blue to the pink or the red to the purple. 1 Mississippi, 2 Mississippi, 3 Mississippi, 4 Mississippi, 5, 6, 7, 8, 9, 10. And sometimes what I like to do when I'm pulling them out is just dip the far corner on the paper towel to get rid of some of the excess dye. Alright, so I've done my stains, and now I'm going to rinse this in water. So we just use regular tap water, and I just turn it on very gently. And I'll keep my thumb right at the top of the water, and I'll actually use that to kind of guide the water down. I don't know, a little too gentle. And this way it'll run gently over the slide down my thumb. I find doing it this way just takes off a little bit. Sometimes I'll lose a clump, especially at the top. Alright, and I'm just going to pat a little bit of the excess off, like so. And I'm going to put it to dry. Here, just for a minute while I do the other one. So I'm just going to dry my hand a little. Grab a paper towel. Do the same thing for the next side. Just touch the corner. Touch the corner. Seven, 8, 9, 10. All right, that is good for those. I'm going to move you closer so you can see kind of how I rinse a little bit better. Okay. try to get my hand here. So I just use my thumb right here at the edge of the frosted slide, so it's right at the top of the slide. And turn the water on lightly. And just try to use my thumb to kind of guide the water a little bit better. Sometimes I find that I lose the chunks right here at the top. I find this is just a little more gentle. Grab this. Okay. And then I'll cap my bottles. Okay, so I'm just going to put these back into our lab area, butt side down, to dry. They should just take a couple more minutes, and then we'll go ahead and read our blood samples. Okay, I just wanted to cut back to the lab just to show you guys our slides drying right there, butt end down, so the feathered edge down. This one's not quite dry yet, but this one's pretty close. So I did end up, my label before kind of washed off the top, so you can't read it very well. So I'm going to relabel this with, oh, I lost my pencil. I'm going to relabel this with the patient's name, Max. He is a dog, and today's date is 6/2/17. And I'll put that back up to dry just a little bit more before we read it. Same with this one. Label it again, Max, dog, 6/2/17. That is good to go. Oh, right there.